Yeah, redfish.
Hagfish.
Hagfish, yeah.
You say it in the
Bridge
same way
nav.
Rennie does, which is sort of displeased. Hagfish are
Can we
cool.
track a line bearing 101, speed 0.4?
Have you ever had to clean up after a hagfish?
Thank you.
All right,
No,
so we're
but I
changing our
still
heading.
think they're
Yep.
cool. I know they make a lot of mucus.
Well,
Well,
clean up after one.
right, I've already said this once, but you may not have room in your heart for hagfish, but they've got 4 or 5 hearts, so they've got plenty of room for you.
This is a Prince star.
Oh yeah. Wonder if they're in the same group.
It's more of a solo artist.
I wonder if they hang out.
Welcome, Miss Escobar's class from Texas.
Yeah, 101, and when we get to about there, we'll do a transect.
Okay.
So
Yeah, I'm hoping that there's hard rock there. If there's not hard rock there, we'll just keep going.
okay.
Yesterday, did you say Hercules is almost like 18 years old?
Yep.
Yeah. 18 years old, Miss Escobar's class.
Yeah, definitely old enough to have a driver's license, but I'm not sure if Herc can vote yet. So I could probably just look at the
VIN number.
I was going to say the metadata on the photos of the build for Herc.
Yeah.
And we're bringing your heading to, what was it?
101.
101?
Mm-hmm.
That's very specific.
Yep. Oh yeah.
But, you have a heading offset. I don't know if you've noticed, but there's a slight difference in what you read versus what is reality. So 101 is in what the raw data of the sensor is a little bit off.
So that's magnetic, right?
Yeah. The crossbow is not magnetic. That's another gyro.
Is it?
I think so. But it's on optical.
I didn't know we had a gyro on Argus. Well, I know the TCM2 is magnetic.
TCM2 we don't like to talk about.
Yeah.
Or no, the TCM2 we can talk about. It's the microstrain we don't like to talk about.
Yeah.
That one's just
Garbage, yeah.
Yeah.
Total garbage.
I like the use of the overlays in ROV nav.
That's going great. For me. Do you want to see an overlay in ROV nav? Let me just fix that for you. Just give me a second here. There you go. You could tell my opinion about that.
Yeah, Rennie's snarky this morning.
How long does it take to reach this depth with Hercules?
This depth, not very long. I've seen a lot of little openings with bacterial mat.
Yeah. Oh yeah, that is right there. Yeah, down to this depth, it would just be like 10 minutes. Not even.
We do an approximate is 1 hour for every 1,000 meters, and right now we are only 163 meters deep.
So by the power of division, just mere minutes.
Aaron, let's push in on the weird bacterial matty looking stuff.
What's the significance of a bacterial map?
Well, in deeper areas, it would indicate that there's some kind of flow, a diffuse flow, or some kind of hydrothermal activity. but this is shallow, so I don't know if it necessarily indicates the same thing. This is photic zone.
Numbers look good.
Awesome.
Yeah, I was thinking that this shallow it might be something that's been long decomposing, and that's
It
just
could
what's
be.
left.
Yep. Yeah, especially in that case where it was just kind of out in the open.
Yeah. Man, these fish just kind of like appear when they zoom in like that.
You can always check out nautiluslive.org to see what technology we use to conduct our expeditions.
So the sea star video
Yeah.
push in. Yep.
It's like a little lavender color.
It's a cool coloration. So that's one of the Velcro ones. You can see all the little spiny things on the top. All right, full wide, and we'll push back out in front in hopes of finding an octopus. Considering that it is now officially, even based on our time zone, International Octopus Appreciation Day, is that correct?
Yes.
International Octopus Day
Ah,
kicks off International Cephalopod Awareness Days. And we should have a Nautilus night tonight.
tomorrow. Is it tomorrow?
No, tonight, October 9th.
Oh, okay.
Today's the
So
Pretty
we-
sure today's the 8th.
Oh, well, tonight it'll be, according to the time on Herc,
Yeah.
UTC,
it'll be
yeah.
tomorrow.
You scared me. I had something to do on the 8th. I was like, what?
What type of fish is that? Is that a rockfish as well?
Which one?
We just passed it up.
There's the little one in the lower.
Yeah, it's pretty safe to assume it's a rockfish.
I think I saw the one
Video
you're talking
pushing.
about, and it was a rockfish.
Haven't seen a holothurian in a little bit. Tube worm.
We have someone in the audience wanting to know, how do you choose what object to collect or organism?
Well,
All right, full wide.
we have a list of scientists, researchers that are studying certain things. So that often makes if it's compatible with what we're sampling and where we're sampling, and the ability for the ROV to collect it, then
Sounds
we'll
like a
certainly do it.
challenge.
I was thinking of Niskin bottles, multiple Niskin bottles versus just one. I wasn't commenting on the pilot's capability of collecting.
So how does this look to you as far as the transect goes? Is this
Not good.
not good?
Yeah, what I wanted was something more rocky, like
Yeah.
what we just passed over, but there wasn't. Oh, is that an octopus? No. No, so let's just keep going towards our
Sure.
spot. Yeah, so we definitely try and make the most out of our ship time and our ROV time by not just one project or two projects, but sometimes a half a dozen to a dozen different projects will be receiving samples from this cruise.
Right, and like last night we picked up the palm frond sponge.
Mm-hmm. Yeah. So those sponge experts, we sent out an announcement, for scientists ashore, as well as researchers that we know from working with them over the years, and they send us a list. Is that one large and cresting sponge?
Yeah.
Oh yeah. Okay, now that comes into view. So they send us a list. We kind of merged them all together. There's a large project within National Marine Fisheries Service that is looking at environmental DNA and really collecting water sample in areas of known and unknown invertebrates, like different species of corals, so that we can, when we find a coral, we can just test the water around it to see what species it is. It takes years to develop the library, the DNA library for something like that. So you collect a specimen in order to kind of calibrate the DNA readouts.
Thank you for that, Jan.
Jamie, since you're probably the best one to answer this, you want to tell people what eDNA is exactly and how it works?
Sure. Can we also get a zoom
On the sea
yeah.
star?
You read my
Yep.
mind. Environmental
Aaron, let's push in.
DNA is, rather than taking an individual organism and extracting the cells directly from that animal, it's getting a water sample and then looking at the DNA within that water. So all organisms are releasing some DNA by shedding skin cells or through some feces or mucus or other components of their bodies that slough off. And depending on what comes off, sometimes it's possible to get DNA from those little particles in the water. And so when you do an environmental DNA sample, you can see a large array of fauna or animals that are in a region without necessarily directly observing those animals and without specifically sampling them. So when you do an environmental DNA sample, you can get an idea of what's around even if you didn't directly observe it.
Jamie, did you work with eDNA at all in your PhD dissertation?
I did not.
What was your PhD dissertation on?
I looked at the community structure of methane seep communities.
It's by the what structure?
Community structure of methane seeps.
Okay.
So the different organisms found at some methane seeps off the east and west coasts of the United States.
Cool. All cold seeps.
Yeah.
This is a nice feature, so let's start at time transect here.
Time transects.
Yeah.
All right, what's our heading exactly?
101.
101.
I didn't hear what you said. I just guessed in context what you were asking.
What are you going to name this, Renee?
Oh. I don't know what do you want to call it.
We're really close to a boundary line. How about boundary 1 or something, because we're not near any waypoints.
Okay.
Or I guess the closest one we're near is 1AEE, but we're pretty far from there by now.
Boundary
I'm
1.
gonna
Okay.
step starboard a little bit to get more underneath you, and then we'll start our transect. We may need to slow the ship down. Oh, you're doing steps. Oh no, you
no,
just
I'm tracking
you just
at
tracked the line.
0.4.
Okay.
Rennie, someone in the audience would like to know, what is your favorite sea creature?
Yeah, well, whoever's asking that probably knows the answer already. I'm going to change it, you know. It's no longer this flamboyant squidworm. It's now the deep starria jelly.
All right, starting transect. Jan, I'm going to fly the altitude manually and just try to stay as low as possible.
Okay, thanks.
Please keep checking out nautiluslive.org for the 2020 expedition. Mr. Dwayne from North Florida, thank you for tuning in.
There's sadly like a very long list of people who could be asking that question.
Why sadly? You have all these people who care about
Yeah.
what your favorite
Nobody asked me what my favorite like map is or anything.
Oh,
Rennie, what's your
Gary,
favorite
if you
map?
see something, we'll have Rennie drop a target. We can come back.
Yeah.
What's your favorite map?
I don't have one. That was a poor example.
Oh.
Couldn't think of anything on the spot.
What was Jamie? What was the question? What's your favorite
Map.
map?
Yeah, Rennie's offended that we always ask what's your favorite animal, and he wants something more applicable to his interests.
What's your favorite remote sensor?
Ooh, okay.
You can do top 3 if you want.
Still hard.
All right, how about remote sensing technology?
Yeah. Multi-beam sonar definitely. Still. I came from LiDAR before this, so those are the two that I like the most.
Guy's back here. Guy, what's your favorite remote sensing technology?
Definitely have to be multi-beam.
Yeah. It's
Okay.
so great. He probably likes the backscatter.
can someone
Nice.
kind of
Okay,
elaborate what multi-beams do for those who don't know?
Instead of
Do you want me
single
to-
beam.
Yeah. You want me to go into the
Oh yeah, go into the
the
details.
whole thing?
There's actually a good description with some animation on
Will
the
you catch back up
OET
into view?
Nautilus Live website.
Probably not. Tilt down as much
Describes
as
it.
you
There's
can.
photos. It's
Yeah, there's a
pretty good.
video on YouTube that I think Clara and Nicole are talking about multi-beam and how it works. And
Yeah, I've done the description here from this seat, and I try my best to not use hand visuals and just like describe it. And it's pretty. Once you get into how the transducer arrays work, it's kind of difficult to express verbally. But I've done it before. We'll
you
wait.
s- you
We'll
saying
wait
you're
till.
gonna do it again.
Yeah, maybe we'll wait till sometime when we're
It's a pretty
towing or
sponge.
transect or something.
That is pretty.
And Jan, I went 0.25 for speed since it was rocky stuff.
Yes.
Should have confirmed that earlier, but
No, when you started, it looked like it was slow, pretty slow.
Go ahead, Lindsay.
so for this, it would have been fine to
So
use
Lindsay's
the
saying that in the photo albums on the website on nautiluslive.org, there should be someone who is doing some daily blogs on mapping that are also pretty informative. There's a lot of, even just our organization, there's been a lot of content that describes what we're doing and why we're doing it as far as sonar mapping goes.
Yeah, it's really hard to decide where to dive and where to head without the mapping data. To me, the mapping data is crucial. You have to do it before you dive, otherwise you're diving blind, and you might hit a rocky area, but the seafloor is, there's a lot less rocky area than there is soft, flat.
Yeah, and satellite altimetry only resolves so far. So really, critical tool in ocean exploration is multi-beam sonar.
Anyway, what I was going to say, from a vehicle standpoint, this is gradual enough in the up and down that I could have stayed, let the vehicle take care of the height off bottom. But if it was rockier, then since the reading's in the back, you can drive straight into a rock with your front porch, which would kind
Jamie,
of
did you
not
capture
be
that
what
we
you
went
want
over
the
the
transect
management
to be.
boundary?
Yeah.
It's okay. We just passed it.
Rennie, how long ago did we go over that boundary?
In time units or in distance units? Because I can only give you the latter.
Distance
Well, you could
is
figure
fine.
out the other one. I'm
15
going
meters.
Okay.
You're
quarter of a
going
meter.
It's going
second.
quarter of a meter per second for 15 meters.
That's what you're saying, Greg.
So we were
is that you should
crossing
just drive
from a
backwards.
closed boundary into a
Open.
open. Is it projected to be closed?
No.
Just open?
Open,
Okay.
I think, to zoom out.
And I could do this off of the
1 minute
altitude number, but I'm really just trying to keep the lighting the same.
How many meters ago was it now, Rennie? Was it still 15 or is it more like 20?
And the higher
Really
off
getting
the
specific
surface
here.
you're flying.
25.
The bluer thing is
25,
good.
okay.
Yeah, but also
Thank
then the easier, you know, the more you can trust an auto.
Right.
altitude.
Yeah, you know, like if I'm flying at like
Roughly.
5 meters off the bottom, then it's very unlikely, unless it's a cliff face, that I'm going to bump into a rock.
Yeah.
But if you're trying to do a meter or a meter and a half, and you're going upslope and you've got rocks in front of you, then you can bump into something. I remembered this time, Rennie.
So a clarification on that. We went from an area that is closed to remain closed, and we're going into an area that is open but proposed to be closed.
Okay,
Yes.
thank you.
Yeah, there's a lot of overlapping polygons on there
Yeah.
of open and closedness.
I believe the ROVs have been in the water for 20 plus hours. Is that correct?
We went in at 4 o'clock
Oh.
this time, so
Oh, okay.
16, over 16.
Do we have a screen that has weather updates?
No. We have a screen that shows what the wind is doing currently. But having
And we have a screen that Shows what the current is doing windly.
And you say I'm the one who's not to be listened to?
Okay, that's around 100 meters so far that we've crossed.
Yeah, I dialed in 200 meters, so.
Cool.
Haven't seen that before. That guy's bright yellow.
Yeah. So bifurcated leg, arm.
It's making a wish.
And then I know Argus's heading on the ROV nav is not correct, but is there, how much do we trust the Herc heading? Like, is there any time delay or?
No, I
No,
think
we trust
dead
the
on.
Herc heading.
Yeah.
Pretty much spot on. If we'll line up on the same heading, and you'll notice that, like, if we're visually on the same heading, then that our heading number is off. Herc's heading is coming from that octane, so it gets it's constantly being calculated. It's a more robust technology. Surprisingly for, you know, fibers and fiber optics.
Yeah.
Guy is pleased with his multi-beam data that was collected. He's saying that it was hard, but not rugose. And that's what we have.
Yeah.
Cool.
Yeah, we're shallow enough that any multi-beam data here would
Yeah, you
be
get
really
good.
really nice.
Actually, it's been interesting since starting working with this vehicle system and this ship before they had the multi-beam. And
2012 or 13 that it was installed.
and the dives that we were on versus how we pick locations for dives now. I think there's still kind of, at least for the kind of seamounts, we go up on ridges more than you go up on a flat. The idea being that you're going to see more life, but there's a real effort to be able to have enough data to basically take a multi-beam map and say, like, okay, you know, this will have more life, this won't. We're working with, what is it, Chris Kelly at University of Hawaii.
Papahanaumoku,
Yep. Trying to figure out what the difference is, and so could you accurately predict the kind of hotspots of life without sending down an ROV to take a look? He gave a really good talk about that when he was on board. Was that last year?
Yep. 150 meters elapsed.
When you can't see anything now because we don't have any stuff, but if there's like whip corals or like some soft things that are kind of sticking up and we're doing a transect like this, you can use bubble to get a really good idea of what your true height is off bottom, because you'll see it kind of bend as it hits the front porch. In this case, it's mostly shadows. We are losing ground, but I think we'll be able to finish.
Yeah.
So we might be getting shallow enough that we could find a. We might be getting shallow enough we could find a fish that liked the laser beams. I keep seeing these big rocks like just off to port.
Is that where we came from?
To port?
Yeah.
No, it's just they're like every now and then, and I've seen them in starboard too. So there's just like there's random big rocks. We just haven't driven over one.
Well, after this, let's drive over one, because we're almost at the point where we wanted to be with this hard flat. And then I think, at least on the nav screen, we can head over to those green pixels.
You know, I just realized something. So I've been typing notes while we've been doing the transect, and I typed some that said like seeing fewer rockfish and almost no rockfish, and I just realized that corresponded actually fairly closely to when we crossed the boundary line from the closed to the open zone.
Is that a crab?
Crab.
So you're saying when we moved into the open zone, the rockfish kind of disappeared?
Yeah, I mean, look around. I've seen like 2 in the last several minutes.
I think
whereas
once we
before,
get up to the rocky bit, we might see
yeah,
them again.
maybe. But I feel like even on the softer flat substrate before, we were still seeing them swimming back and forth. Yeah, before we were seeing rockfish frequently.
All right.
So I think
Where do you
it's
have me
making
as
a difference.
almost done?
Oh, yep, 196. Perfect.
I love it when a plan comes together.
There we go. That's about 200 meters.
And done.
Right.
That's pretty good.
Yeah. I also just dropped that target at random. I didn't measure out 200 meters, and it's right on.
Which way you want to go, Guy? Want to go over to this green pixel?
To the east?
This one or this
A little
one?
northeast?
That one. And then how do I, okay.
Are you on south or east?
East.
East. Cool.
Yeah. So we're going to head over to the light green pixel and go across the green pixels that get darker.
All right. Well, since we are still tracking a line, I'm going to catch up.
Yeah.
Okay.
Go do that.
So has this area been trawled recently? Do you know?
I don't see trawl lines.
I don't see trawl lines, and I tried to get the trawling records, which are actually a little more difficult to get the actual locations, and I could not before the cruise.
Okay, so do you want me to stop the ship, and we're kind of going... You said you wanted to go east from here?
Well, it was just we're going. There was a little bit of
okay, and then we'll go
going that way.
And then we'll go north and then over.
What we'd like to do is to go over to those light green pixels.
Yeah, here, got it.
Lower.
Here.
And then go across. So start in the light green, go across, kind of investigate this area up to 1523.
So go up like that?
Yeah.
Okay. Then we'll
Might
continue on this track,
yeah.
and then we'll go up.
We want to kind of hit those darker green areas.
So through up like that.
Or zigzagging across.
Or like this and then up. Okay, so bridge nav. Let's hold position. And can we have a ship move bearing 04040 meters?
All right, Aaron,
Thank
give it
you.
everything you got.
So I'll step up to here, and we'll go east a bit and then up. Cover as much of that as we can.
And I think I saw a C-pen this way. Aaron, come out a little bit. There it is. All right, push back in.
Yeah. Another one of those Virgularias. Yeah.
Yeah, tiny.
It's going to, if we touch it, it's going to sink in.
Is this one with the kind of muscular base that pulls
Yeah.
itself in?
Uh-huh.
We could try to dig it out from the bottom and come on that way, but if you're interested in it.
No.
Okay.
That's okay.
Full wide.
They're really small and they go really deep, and it's just really hard to sample them.
All right.
Yeah.
Renee, I was distracted by a shiny thing. What direction are we going?
Just going 040 for a couple steps.
Okay.
We'll get up to the green bit, and then we'll go east.
Yeah, I'm going to check
we'll go
my
up.
specimen request
Over here.
list and see if that Vulgularias are on there. Could be a challenge for you.
I'm all about the challenges.
A lot of brachiopods here.
Yeah. There's another crab. And we have rockfish again. Yeah. All right, Aaron, let's zoom for Jamie.
Thank you, that was lovely.
Little fish. Okay, full wide. Crusting sponge.
Bridge nav. Add one step.
All right, Aaron, let's push in. Look at that tube.
Hmm.
On a little sea star. All right, full line. That curls all the way around that rock.
Mm-hmm.
Wow.
Guy's been around for a while.
It's been around for a while. Losing it.
You say that a lot, and I don't know that you know what it means.
You don't know that I've ever had it.
You have to have had it. All right, so we're still short of jellyfish and octopus. Aaron, jellyfish and Argus.
She's not satisfied with those. She's very picky.
Yes, I am.
We appreciate that about you. It probably makes you better at your job.
Thank you.
If we see an octopus and Argus, I'd be really impressed.
So we're going to want to go east from here? East and maybe a little south.
Yeah.
Okay.
Kind of just explore this region. We'll zigzag. We'll go east, but we can. Hercules has some tether to go either way.
Yeah, zigzagging's good.
Okay.
And it's also good to like get to the base of it and go up slope to see the full range of habitats.
Yep, we can do that too.
All right, so let's bring your heading east. Almost, we're almost done with that ship move.
Yeah. Bridge snap. Could we have a step 40 meters bearing east? Thank you.
Trash fish.
Trash.
Aaron, push in.
Trash mat.
Trash mat.
Yeah,
To trash bath mat.
Trash bath mat.
fish.
Or is it in a, yeah, it's a
Yeah,
bath
it's one of
mat.
those
Bath
bath
mat.
mats
Yeah.
with the little adhesives on the bottom.
At least we know if we were standing down
bottle.
there, we wouldn't slip.
Oh, hang on.
It's a rare planar octopus.
Planer.
And a bottle.
Did you say and a bottle?
Yep. Or
Something.
Oh, yeah.
something too angular to likely be natural.
Maybe it's
Full line.
shampoo.
Well, they're all about the hospitality in the deep sea.
The fish are just trying to keep clean.
Rock.
Always fascinates me when there's just like a random big rock.
I have no explanation for that either.
Yeah.
What's, can we zoom in on this,
The little guy?
yeah, the juvenile,
Video push in.
is it a what is it? It's a juvenile
Hold that.
yellow, no. Is it a juvenile yellow one? Help. Erica, Tom. I think they're so pretty.
Oh.
Oh, he went into his hidey hole.
All right, Aaron, full line.
Is that an octopus?
There, you see an octopus?
I saw tentacles coming out of the hole.
Oh. If it is, it lucked out. It is.
Oh, zoom in on that little pink thing. Oh, never mind. Never mind. It's just fluff. Pink fluff.
Okay, passing the pink fluff.
It's crinoid rock right there.
Yeah.
Yellow eye, thank you.
That's the one they want.
Really, that becomes a yellow eye?
Yeah.
Huh.
Well, there's a lot of crinoid up here. Look at that.
Oh, that one rockfish, its tail was all bitten off.
Bridge nav. Let's do one step, 40 meters, bearing 135. Thank you.
So we're just going to kind of slide this way, but we don't need to bring our heading around.
We'll just get down to the base of this
Yeah.
and turn around, come back up. Different way.
Are the white lines in this map always 10 meters?
No,
Or
I
does
mean, we
the
can, well,
scale can change?
no, the scale doesn't change. They're always 10 meters. They're static here, but we can make them different on a different dive if we wanted.
Cool.
Yeah, it's always best to ask.
Yeah.
Hey everyone, Timmy wanted me to let everybody know he loves you all. I don't know if y'all know who Timmy
Thanks, bud.
is. Someone asked a question about how normal is it for fish to just sit there like being stationary? They were under the impression that movement was essential.
Not all fish need to move in order to breathe. There are some sharks that need to have water flowing over their gills to get enough oxygen in, but a lot of deep sea species, though we're only moderately
Wrap
deep,
in the
but
cable?
a lot of deep sea species are fairly sedentary to conserve energy, or they swim very, very slowly to conserve energy because resources in terms of food are scarce in deeper water.
Yeah, some of them lie in wait. They're ambush hunters, so you see them not moving at all, but that's their way of just hiding and waiting for some food to come by, and they can reach out and quickly grab it.
Can look left a little bit if
Just doing a minor pause in science operations. We check out a engineering issue. That's what we're discussing here.
We are currently on Nautilus.
I
Thank
think
you for
your
tuning in.
end. Oh no, my end is over. Well, shoot. Okay. Like, okay. That increased my delta. Ooh.
You can find more information about the Amelia expedition on nautiluslab.org.
Oh, it looks like, did it come undone? It looks like it came out. And tighten it, get the daisy.
How high should I come up? 35.
Ship move is stopped, so. Yep.
Okay. At 35.
Greg, are we on deck at 12 or 1 today?
You're not on. Yeah, it depends on the weather. If it's still fine, then we can stay in a little bit longer because there's no time to surface. It's very minimal, so.
Tether tug. Okay.
If things are still looking good at 12, then stay in a little longer. So far, we haven't really seen too much.
So
The
let's
wind's
bring
around
your heading
17, steady.
back. Let's continue going around that way.
Okay.
Yeah. And I'm going to come back towards you to give you that ability. Wow, look at all those fish.
But we're still tugging. Should I come down at all? Oh.
I'm going to come back towards you
Okay.
first. And you can come down to 25.
You're also here.
Yeah.
I'll reset that once we get settled.
Do you want me to come over to 135 or 135?
Yeah, I think we're going to actually end up going
north, roughly.
All right, but that looks a lot better.
Yeah. It was that football was the issue? Okay.
Yep, that football was the issue. Okay, so we're going to end up going north?
Yeah, somewhere around there. Is that what you want to do, Jan? We'll go kind of up this slope. If we're starting at the base, go up.
Yeah.
So I'm going to drive around the long way.
Let's cover some different ground and we'll go 010.
And you can follow me around back to north.
Clockwise or counterclockwise?
You would be going clockwise.
Someone on our live feed would like to know, do coral and sponge have a nervous system? And is that how they feel particles in the water they feed on?
Well, that's a really good question, but I don't have the answer. Anybody else? Gary?
Well, sponges don't have a nervous system, but corals have a very primitive nerve net, and they can transfer information from polyp to polyp, but it's not like a brain. So they do detect food mostly by chemical senses and tactile response, meaning if something touches a tentacle, they can grab it, but that's about it.
Do you want to hold for a few seconds? I'll do a reset.
Thanks for sharing your question.
Just a couple.
So Gary, I've been seeing a lot of what I've been in my head calling pink fluff, and I don't see any in the screen right now, but I want you to take a look at it when we see it. It just went
call
out.
it out next time. And
Yeah.
Get a close-up of it. We'll have a look.
I've been thinking that it was like
ready?
Ready.
Good.
some type of.
Was it just out of screen?
Yeah. Well, we'll see more. I'm pretty. Oh, there it is. Lower screen. To the right of that sponge. There's one.
Coral, is that what we're talking about? The red?
I don't know.
Video push in?
Just kind of flapping
Because
around.
that's a coral.
There you go.
There's
Okay.
actually 3.
Yeah.
Some of it's not looking too healthy.
So we've actually been seeing these here and there, and I've been thinking because the polyps are so. We collected one of these last night, right?
Yeah.
Yeah. You want one more specimen? Yeah. You go over to the left, underneath the sponge. Oh,
Hang on, too far. There's a very little one.
that's a very, very little one. This is gonna be, yeah. There's 3 of them there, and they're not numerous, but they're here and there. And I've been thinking, oh, it's just a pink fluff. This is my pink fluff. I think the one over, this one's redder than the one to the right. Do you want this one, or you want the one to the right? I think is going to be easier.
Ship holding.
Yeah, Greg, whichever one is easiest. I don't think the one on the left is going to be easiest. I think that one. Yeah. What about this siltation on it? Is that just like a little injury or something? Something's growing on it. Where?
Aaron, let's come full wide.
Yeah. Because Gary's thinking that it's so different than something else he's seen that if it is a new species, check out the variation.
Yeah.
Also helping to describe it.
If we went for the super little one, we could do a slurp and scrape. If we went for the bigger one on the right, then we would do a snip.
Yeah, snip is close to the bottom of the larger one.
Okay.
Because the one we got last night was pretty small.
Yeah, it was really small.
Yeah.
All right, I'm going to bump forward a little
No,
bit.
I've seen from a distance, it just looks like a little fluff. When the polyps are out, it looks very fluffy. And until we actually stop, but I've been seeing them along the way. I went, what is that pink stuff?
Yeah, they're so tiny.